Advances in warm water fish research. Aquaculture. Aquaculture is the production of vegetables or animals in fresh water or salt water. These are used to feed humans or other animal species, as well as for the production of raw materials. Currently, this practice accounts for 25% of the protein consumed around the world.
Among the benefits of aquaculture are the increase in fish production tonnage worldwide. When fish reproduce naturally with their environments in saltwater or freshwater locations, the truth is that they don't reproduce efficiently, nor do they have high survival rates. What has been achieved with aquaculture is to increase production in terms of tonnage, in the number of growth cycles, in the number of offspring produced throughout the year. We have increased their survival rate. We have also even managed to increase their size. We've improved their conversion rates, all of it as a result of trying to produce fish under the most ideal conditions from the point of view of the water's physical and chemical parameters, focusing on nutrition, on infrastructure, on genetic management, on adequate management. The idea is to increase production to the optimum point.
In Colombia, there are organizations with researchers whose objective is to study issues related to the biology, reproduction, nutrition, feeding habits, and production of aquatic species, native and exotic. Those that have biological and economic importance to regional and national development plans in the ornamental as well as the food sector.
Como ornamental.
Laboratory.
In this lab, we are currently conducting some parts of the research, given that the rest is done in the fish farming station or in the molecular process laboratories. Right now, the lab is producing live food, Daphnia magna, as well as Daphnia pulex. Likewise, we are producing another type of living food that is called a microworm, Panagrellis redivivus, and we are conducting trials to standardize the production methods to find the most appropriate ones, those that can prolong the life of the crop, with or without enrichment. We use pigments to dye the living food so that it is easier for the fish larvae to visualize and eat.
This lab has 50 aquariums. Each one has a thermometer, as well as aeration and heating equipment.
Having such a large number of aquariums has allowed us to develop various treatments, and each treatment is repeated several times so that our results are reliable.
Life cycles of an aquatic organism.
First comes the egg phase. When fertilization takes place, it happens during the ovum stage. It undergoes an incubation process that takes a different number of days depending on the species. In some, it takes 18 to 24 hours. In other species, it takes 3 or 4 days. In other species like trout, for example, it can take up to 28 days. So after the incubation period, they hatch. After it's born, we call it a larva.
The larval stage is a stage in which the fish lives from its own corporeal reserves, meaning the yolk sac.
The time that the yolk sac lasts is called the reabsorption of the yolk sac. Some species reabsorb it in 1 day. In other species, it takes 4 days. In other species, it takes 10 to 15 days. Once the reabsorption finalizes, then it starts to feed itself, either on living or inert food.
Inerte o alimento vivo.
Living food is a type of food that the post-larva consume for the first time. Not all species are capable of eating inert or dry food, meaning commercially produced feed concentrate. For some species, it is necessary to eat live food for the first time they feed. It needs to contain aquatic organisms, be it of vegetable origin like algae, microalgae, or they can be protozoan or infusoria. but they have to belong to the animal kingdom, like rotifers, copepods, cladocera, or brine shrimp.
Artemia salina.
Why must they be fed live food? When species need to feed for the first time, they cannot consume inert food, either because of the size of the particle or because it's not sufficiently attractive to the post-larva and it won't be stimulated to seek it. Living food is a resource of great nutritional value for ornamental as well as for species eaten by humans. The main reason lies in the fact that these organisms constitute a nutritional capsule that usually contains the basic elements of a balanced diet. The main advantage lies with the fact that they keep their nutritional value up until the moment of consumption.
Living food is not needed throughout the entire life. Approximately 3 or 4 weeks after having completed the yolk reabsorption, you can administer inert food to those species that tolerate it, or you can begin to feed them what we call forage fish.
Daphnia magna. The Daphnia genus is a microcrustacean that belongs to the Cladocera family, Daphniidae. It lives in freshwater and in saltwater. Its size varies between 0.2 and 5 millimeters in length, and it is used as living food for different stages of the post-larva.
Anatomically, it's an animal that has a head, a stem, and legs, with which it moves through the water. Its body is gelatinous, transparent, and biologically it reproduces sexually as well as asexually. In this type of setting, its reproduction is basically asexual, meaning that in asexual reproduction, we obtain over 90% females, and they're the ones that provide the final Cladocera population.
Maintenance of this type of culture is very simple. To start off, you have to clean the walls of the aquarium. Afterwards, in order to avoid harm to the Cladocera, with a very thin hose, we vacuum out the organic material that sediments. And to finalize, we replace 30% of the water in the fish tank. Panagrellus redivivus. Another type of living food is the nematode Panagrellus redivivus, a white or transparent, non-segmented microworm that can reach up to 15 millimeters in length. These microworms are live-bearing and they reproduce sexually. You can differentiate males from females. The female has a tail with a pointy end, while for the males, the tail end is shaped like a hook. Also, males are present in much smaller proportions. They are smaller and more robust. Growing Panagrellus redivivus is easy and very economical. We only need a vessel, commercially available oat flour, and water. The procedure is as follows. In a 15 by 10 centimeter vessel, we are going to weigh 60 grams of flour and we will add it to the vessel. To the flour, we will add 80 grams or milliliters of water, and we proceed to mix it. The ideal is to get a mixture that is more humid than dry to ensure adequate survival of the microworm. After mixing the oat flour and the water, we proceed to inoculate the mixture with around 2 or 3 grams of microworms from a previous batch. 5 days after the mix is set, we will get something like this. To proceed to feed the animals, we use any piece of cardboard and we scrape off the worms on the edge of the vessel and we place it in a small PVC tube with a filtering mesh so that we can clean off the oat flour and any other debris. The nutritional position of the Panagrelus redivivus is influenced by the cultivation medium. The literature reports the following: between 76 and 78% water, 22 and 24% dry matter, 40% protein, 20% total fat, free nitrogen at 40%.
40%.
Artemia salina. The Artemia salina is a microcrustacean that is well known in aquaculture. It's used as a live food for post-larvae.
Artemia salina is normally obtained in the marketplace in 454-gram cans. And what you do is to seed the Artemia salina so that the 24 hours after seeding they will hatch. At that point, the process of seeding the Artemia eggs is very simple. You use at least 1 or 2.5 grams for every liter of water, and you add 35 grams of sea salt. You aerate up to 100% saturation. You keep it at a temperature of 31 degrees Celsius. And we hope that the batch of Artemia will hatch 24 hours later and become a nauplius, which is one of the forms of the post-larva, one of its stages. Then that nauplius will be mobile. Their color is more or less brown, and they are easily spotted by the fish. Does not work for all post-larva because it's not the smallest in size, but it has a good nutritional value. Now, Artemia nauplius can be around 42 to 45% protein. It has a good amount of polyunsaturated fatty acids, and it also has sufficient carbohydrates.
Ornamental fish. Among the different types of work being done in the lab, we are conducting research on ornamental fish. What we are trying to do with them is not just to standardize reproduction, but also to conduct research on their nutrition and feeding habits. Through our work, we are conducting research with live food and non-conventional food like pastes, as well as dry feed concentrates. Research is being performed on Amazon cichlids such as angelfish and the banded cichlid. Also, live-bearing species like the guppy, swordtail, and other well-known species like goldfish and Siamese fighting fish. Angelfish. Habitats: Orinoco, Putumayo, Amazon, and Guaviare rivers. Among the morphological characteristics of the angelfish are that it has a disc-shaped body, flattened on the sides, with a small head. Its front nasal region is concave. Those are its main traits. It also has a very small mouth. According to the literature, males are physically different from females. Their front nasal regions are different. The female's is concave, and the male's is convex. Physical and chemical requirements. Average water temperature of 25 to 28 degrees Celsius. Slightly acidic pH between 6.5 and 7. Soft water with abundant organic matter. In the lab, we try to feed these fish with a diet that is as close as possible to what it would be in their natural habitat, such as chironomids, brine shrimp, and water fleas. They also adapt to concentrated feed and some non-conventional homemade pastes. Heros severus, or banded cichlid. Habitats: Amazon, Orinoco, Vichada, and Inírida rivers. The main morphological traits of the banded cichlid are short head, disc-shaped body, small mouth, and thick lips. Males are differentiated from females by straight red lines visible in the forehead. Physical and chemical requirements. Average water temperature of 25 to 28 degrees Celsius. Slightly acidic pH between 6.5 and 7. Soft water with abundant organic matter. The diet of the banded cichlid, Heros severus, is very close to that of the angelfish. In their natural habitat, they feed on live prey, such as chironomids, mosquito larvae, water fleas, and brine shrimp. Swordtail. Habitats: Central America, Mexico, and Guatemala. The morphological characteristics of the swordtail are very similar to the guppy. They have a flattened back, a bulging belly, and an upturned mouth. The size of the females is on average 12 centimeters long, while the males average approximately 8 centimeters. They are reddish in color, and the males have tail fins shaped like a sword, which sets them apart from the females. Physical and chemical requirements. Average water temperature of 22 to 25 degrees Celsius and a neutral pH. The diet of the swordtail is based on live prey, very similar to that of the guppy, using brine shrimp, water fleas, and mosquito larvae as a food source. They also adapt well to feed concentrate and nutritional paste. Goldfish. Habitat: Asia. The morphology of the goldfish is varied. Depending on the species, they have varied head shapes, sometimes lion-shaped. Also, they have different types of eye shapes, some like a bubble or a telescope. Physical and chemical requirements. Average water temperature of 22 to 25 degrees Celsius and neutral pH. The goldfish diet is primarily plant-based, and they adapt very well in captivity to eating feed concentrate. Siamese fighting fish. Habitats: Thailand, Malaysia, China, Cambodia. The morphology of the fighting fish is they have compact bodies, upturned mouth, and thick lips. Also, their fins are long and colorful, which makes it a very striking fish. Males are differentiated from the females by their size and tail shape. Males can have tails shaped like a half moon. Physical and chemical requirements. Average water temperature of 22 to 25 degrees Celsius and a neutral pH. The dietary habits of the fighting fish are very varied. They primarily eat live prey, such as water fleas, chironomids, and mosquito larvae, and they can adapt to feed concentrate. Fish farming station.
This fish farming station is located in the village of San Jose del News, in the municipality of San Roque. The fish farming station is dedicated to teaching, research, and training. It's a warmwater station, and therefore we only handle warmwater species in this location. Among the research being conducted here, we are fundamentally focused on all aspects of induced reproduction of native species, as well as in growing post-larva, which are very difficult, if not impossible, to grow in captivity.
Validation of YY supermale tilapia production.
We are now extracting blood in order to be able to perform PCR tests, and so we can determine which primer to use in order to distinguish the sexual chromosome of these fish. The ultimate goal of our research is to be able to produce tilapia populations that are genetically male, meaning 100% XY chromosomes. Nowadays, throughout the country and throughout the world, people produce monosex populations, not genetically, but phenotypically. They supply a hormone along with the post-larva's first feeding for a period of 21 to 30 days. This is a masculinizing hormone, and what it does is to convert the gonads of genetic females into testicles. And this has the effect that nearly 100% of the population being produced will be phenotypically male, even if genetically they are still females.
We are in Earth Pond number 9 at the fish farming station, where we keep some of the reproductive units we use to obtain red tilapia eggs. Reproduction takes place in the following way. We introduce females in a 3-to-1 ratio for each male. The male will build a nest at the bottom of the pond where the female deposits the eggs between 200 and 1,000, depending on the age and size of the female. Afterwards, the male will fertilize the eggs and the female will place them in her mouth where she will incubate them. The eggs hatch between the 4th and the 6th day, and the larvae remain in the mother's mouth for another 6 days after hatching, meaning they will have remained 12 days in the mother's mouth. After the 12 days, we begin to provide food with the hormone added to start the sexual reversion process. After supplying the masculinizing food, we take some larvae and we take them to a research pond. There, they will undergo another hormonal treatment. We are now in the station's research ponds, which we use for the Super Male project. We use them to conduct the reversion of some larvae with 17 Betta estradiol feminizing hormone in order to obtain XY neo-females, which will make it possible to obtain YY supermales, and, later on, the genetically male lots. Once we obtain the XY neo-females, they are tagged with different colored tags placed near the eyes. We then conduct progeny testing, where we evaluate the proportion of males to females in the sample. This is an alternative to molecular identification by markers in the lab.
Por marcadores moleculares en laboratorio.
After we select the best females that have undergone feminization, we conduct the progeny tests. That consists of taking one female for every male and analyzing their progeny. So, if 50% of the progeny is female and 50% is male, we assume it's a normal XX female. But if, on the contrary, 75% of its progeny is male and 25% female, then we assume it's a neo-female with an XY genotype. We expect that out of 75% of the male population, 25% will be YY males and 50% will be normal XY males.
Normales XY.